Hello, everybody. Today we are going to talk about the first step of cell respiration, and it is called glycolysis. So glycolysis is that very first step. And we're going to be talking about the steps of glycolysis today, where it happens, and what exactly the process is. As we learn today, don't forget the main point. Everything we're talking about for this entire chapter is to make ATP. That's the point. We want to make ATP. We want to make as much ATP as possible. And this is the very first step in that process. So glycolysis, the lysis refers to breaking. The glyco refers to glucose. This is the process of breaking up glucose. Right? And what's actually going to happen overall, the entire process is basically this right here. So glucose has 6 carbons, and what we're going to do is we're going to break this molecule in half to make 2 pyruvates. Each pyruvate has 3 carbons. So notice we don't defy the law of conservation of mass. We're just breaking glucose in half, and that is essentially what's happening. So keep that in mind. This is what's going on. We start with 1 6-carbon glucose, and we end with 2 3-carbon pyruvates. This is a very ancient pathway which harvests energy. Every single living thing that we know of can undergo glycolysis. It does not matter how simple or complex it is. It does not matter where we find it, in the ocean, on land. Every single living thing undergoes glycolysis. So it is a very old energy harvesting pathway. So this is probably one of the very first things that evolved in order for life to exist. This is where those very first energy transfers evolved, and they are found in every living thing still. So there is not a single living thing in existence that does not undergo this process. Glycolysis is, on its very basic level, a transfer of energy from organic molecules, that would be the glyco or the glucose, to make ATP. And every living thing uses ATP as well. This is the starting point for all cell respiration, regardless of what type of organism you are. However, it's inefficient. It's not great. It's not the best way we have, but it is the oldest way. Glycolysis only generates 2 ATP for every glucose, and that's not a lot. 2 ATP is not very much. We can actually do much better with some more efficient processes that evolved later. All right, so there we have a soccer player. If you can see the picture, that is not enough ATP to actually do all the things that an athlete or any human would need to do. Glycolysis occurs in the cytoplasm. And this is another important thing to remember because glycolysis is old. It predates the existence of other organelles, so it takes place in the cytoplasm because it also happens in prokaryotes, and prokaryotes only have a cytoplasm. They don't have other organelles where this can take place in. So regardless of whether you're a plant or an animal, glycolysis takes place in the cytoplasm because it is so old. So we already answered the penguin's question about why that makes evolutionary sense. So prokaryotes were the very first cells. They had no organelles. They had an anaerobic atmosphere. They did not have oxygen around. And so all life on Earth evolved without oxygen. Energy still had to be captured. Even though there was no oxygen, energy still had to be captured in the absence of oxygen. And so prokaryotes evolved a process like glycolysis, and they are the ancestors of all modern life. So we have this very old pathway that does not require oxygen. It is anaerobic because of evolution. All cells utilize glycolysis. So what the penguin is saying up here is pretty important. The enzymes of glycolysis are well conserved. And so conserved means sort of unchanging. They haven't changed that much because all cells still use them, right? They're very old. They're still being used. So we would expect the glycolysis enzymes today to look very similar to the glycolysis enzymes from before. Okay. So let me move this out of the way. Okay, so how is energy in glucose captured during glycolysis? So here's the process. We start with glucose, and the very first thing that has to happen is we have to add 2 ATPs to glucose. So what ATP is going to do is it's going to phosphorylate the glucose. So we're going to be adding a phosphate to each end of glucose. And if we get rid of a phosphate, ATP turns into ADP. Now you might be wondering, what does this little curved arrow mean? This curved arrow here means that 2 ATP are going to do some work on the glucose, and then they're going to leave as ADP. So they're kind of going in, doing a little bit of work, and then going away. Kind of like if you were to hire a plumber to come to your house, do some work, and then they leave. They change your house, but then they go away. Just like the ATP is going to change the glucose, and then the ATP is going to go away. So the glucose becomes this other compound. You do not need to memorize this name. And this compound is basically going to break in half. So glucose has 6 carbons, so each of these now has 3, and some things are going to happen to each of these compounds. So we break it down into 2 steps: glucose activation up here, and then the big energy harvest at the end. So glycolysis has 10 reactions, and we're going to be simplifying all of them into just a few. Overall, we're converting glucose to 2-pyruvate. First, by phosphorylating glucose by adding in the 2 ATP. So notice what happened, right? Glucose just got 2 phosphates there. So now we've used 2 parcels of energy, and we have this very unstable compound. All right, this very unstable compound is going to break in half. So now we have 2 3-carbon compounds. Each one has been phosphorylated. Okay, these 2 things are called G3P. And eventually, a series of reactions is going to turn G3P. There's a bunch of steps here. We're just going to ignore those steps for right now, and we're going to turn it into pyruvate. Pyruvate is a 3-carbon compound. There's a little bit more to it than this, but we're going to keep things simple. So just know that pyruvate is 3 carbons. How do we do that? Well, we are going to collect some electrons from G3P. See what happened there? NAD plus came in, stripped some electrons away from G3P to make 2 NADH. What can we use NADH for? Well, NADH is an electron carrier, which means it's holding energy. All right, then what else happens is that 4 of these ADP come in, and they take away phosphates. So we had 2 phosphates already here, and then 2 phosphates ended up getting added just from the cytoplasm. So these 4 ADP take those phosphates, and we make 4 ATP. So we've made energy here with the NADH, and we've made energy here with the ATP. So we produce 4 ATP and 2 NADH. Both of these are energy-containing compounds. These have lots of energy. It consumes, though, 2 ATP, so technically the net yield is only 2 ATP and 2 NADH. So we've got energy in the form of ATP, and we have energy in the form of NADH. And we also still have pyruvate. Pyruvate actually has some chemical potential energy sitting there as well. What this is showing you now is that there are enzymes that are going to catalyze each step along the way. Okay, so glycolysis summary. First of all, there is the endergonic part of glycolysis where we invest 2 ATP. Okay, think about that like activation energy. We lose 2. We're spending a little bit of energy. And now we've got the energy payoff, or the exergonic, so we've invested some energy. Let's see if we get paid back. We get paid back 4 ATP with a net gain of 2. So even though we invested some at the beginning, we got double our ATP back, and we also got a little bit of NADH. Keep in mind, this is not much. A little bit of NADH and a little bit of ATP. So the net yield is 2 ATP and 2 NADH. So a way you can think about it is a little bit of ATP investment at the beginning releases NADH energy and releases even more ATP energy. And we're still left with pyruvate, which has energy too. So we invest some energy at the beginning and we get paid back a little bit more. Electron energy. So let's even write that down. This is energy from electrons. This is ATP energy. And down here, the pyruvate is chemical potential energy, still waiting there in the bonds of pyruvate. We can still use that. This is showing the first half of glycolysis. You can actually see that it's a pretty complicated series of reactions. We don't really need to know all of these reactions, but you can see some things that you may recognize. ATP being invested in 2 different places, and you can see here that the glucose is being split into 2. And then the energy harvest phase, we have these 2 G3Ps that are going to help produce some NADH, as you see there. So electron energy, and we produce some ATP at the end. And we call that substrate level phosphorylation. I would highlight this term, substrate level phosphorylation. The phosphorylation refers to making ATP by phosphorylating it, by sticking a phosphate on. And that happens with the help of an enzyme in a chemical reaction. That happened there, substrate level phosphorylation. Don't forget that term. All right, and that is how we get our ATP. So what exactly is substrate level phosphorylation? So substrate level phosphorylation just means we have an enzyme that's going to add a phosphate to ADP to make ATP. So that's going to happen in a chemical reaction. So as part of that chemical reaction to turn PEP into pyruvate, that chemical reaction is going to facilitate the making of ATP with the help of an enzyme. Okay, the phosphate is transferred from the PEP to the ADP with the help of a kinase enzyme. That is something you should know, and that makes ATP. Okay, so the phosphate comes directly from the substrate. That's why it's substrate level phosphorylation. Okay, so energy accounting. The net gain is 2 ATP and 2 NADH minus the investment, and we get a small energy return. And we still have the pyruvate, which is a sugar. All right, so all of that work, and that's all that we get. It's not very much, it's not very efficient, but it does give us something. The glucose, it's been broken in half, still has so much more to give. So that's not a lot of energy, and for 1 billion years, this is how life on Earth survived. There was no oxygen, there was slow growth, there was slow reproduction, and only a 3.5% payoff for that energy. However, there's more carbons to strip off, and that means there's more energy to harvest. So glucose to pyruvate, a very simple pathway, and it's kind of wasting the energy that's still there in pyruvate. So this is what we just did. That is a picture of glycolysis. All right, and there's our abbreviated showing what glycolysis does. So eventually what's going to happen is we are going to run out of NAD+. If we need glucose and we need ADP and we need NAD+, eventually we're going to run out of NAD+ because we're going to keep making NADH over and over again. So what are we going to do about this issue? We need another molecule to accept the H from NADH to regenerate NAD+. So NAD+ is free for another round. So how does that happen? Well, in aerobic organisms like animals and plants, this on the left happens. This is normal cell respiration. But for some bacteria and for some organisms that cannot use oxygen, a process called fermentation has to occur. One of these two pathways can happen. We can't do both, though. Every living thing does glycolysis, and after that, we either move to fermentation or aerobic respiration. Which path you use depends on who you are. So we are going to get to this next time when we talk about fermentation and the next steps in cell respiration.